Perfect. Can everybody hear me okay? I'm actually just going to hold this so it's close to my mask. So hello, everyone. If you don't know me, my name is Bonnie Brzozowski. I'm a reference librarian here at the Corvallis-Benton County Public Library, and I'd like to welcome you to today's Random Review. Thank you so much for being here. So along with my coworker, Mike Hansen, we're the co-chairs of the Random Review Committee. This committee is a group of about 9 of us who come together once a year. We're actually going to be meeting again in May to select books and reviewers for this 10-month series. So thank you so much to the committee and to the Friends of the Library who make this event series possible. And a special thanks to Gary Whitehouse for writing our press releases. If you're not familiar, I'd like to mention our Random Review webpage, which you can access from the library's homepage. Actually, Virginia conveniently has it on her slide. That Random Review button is on our homepage. It will take you to the Random Review webpage. On that page, you can find the Random Review schedule of events, a list of books related to the books that are reviewed that's compiled by our volunteer, Gail Gerdeman, and it also links to audio recordings of each of these events. The audio recordings are posted with transcripts on the library's YouTube page, so you can also check there as well. February's review of Remarkably Bright Creatures will be posted very soon. I'm sorry, we actually had a brief power outage yesterday. I don't know if anyone else experienced that, and the person that was doing the subtitling for that lost all their work. Or else it would have been done by today. So sorry for the delay. It may be done by today or tomorrow. So I'd also like to thank our Friends of the Library group for their co-sponsorship of this series, as well as just their support of Random Review and the library over the years. If you haven't heard, they not only founded and ran the Random Review series for 30 years, they also coordinate and run book sales, which they have one upcoming. I believe it's April 19th through the 21st. Does that sound right, friends? Great. So be sure to hit that book sale. Because those funds benefit the library and contribute to programs like Random Review. So they're very, very important to us. You can learn more about the Friends. You can also volunteer with the Friends. Go to friendsofthecbclibrary.org. Okay, I want to bring us to our main event. Today we've invited Virginia Weiss. She's a University Distinguished Professor of Integrative Biology at Oregon State University. She's going to review today the critically acclaimed book, The Song of the Cell, An Exploration of Medicine and the New Human. And first, I'm going to invite Dr. Barbara Taylor. She's a veteran of Random Review herself, has done a few of these herself. She's also a longtime colleague of Dr. Weiss, and she's going to be introducing us to Dr. Weiss today. So come on up.
Perfect. All right. Thank you so much. I appreciate that introduction, and I'm so delighted to introduce Virginia. As she said, she's my best friend and a colleague since she arrived at OSU in 1996. She's had an amazing research career. She's established an incredible program, trained lots of students, undergraduates, graduate students, postdocs, developed international and national collaborations. She's been recognized for her contributions to teaching and research at OSU by a number of awards, including her awarding of a University Distinguished Professor, as you just heard, in 2019. She has been awarded the Eminence Award for Research by her research community, the Choral Community, the International Choral Society. And last year, she was elected a Fellow of the American Association for the Advancement of Science, a really distinguished career. And I also want to say she's so generous with her time. She served many administrative roles at Oregon State, also service. She was my department head for 9 years. Can't be an easy job, and she kept her research career going at the same time. She served to develop and organize meetings for her colleagues and her research community. She has done it at all levels, and so she's an awesome person, and she's a great researcher. And her research since graduate school has been focused on the symbiotic interaction between individual algal cells and their cnidarian hosts, sea anemones and corals. And she'll talk a little bit about that, and I don't want to steal her thunder. But what I think is so important about what her research program was is that just like they develop a mouse model for humans, because humans are notoriously hard to study, you can't do the kinds of experiments you need to do. So Virginia and her lab developed a little lab model called Aptasia, a sea anemone off the coast, so they could study in great detail the cellular and molecular regulation of symbiosis and how it breaks down when symbiosis goes bad. And corals ultimately will be able to understand corals from that. Virginia, I'm so excited to hear about your...
Okay, thank you so much for that lovely introduction. Barb is retired, and so I miss her. I don't see her daily, but we're still close friends, and I thank you for that. Okay, so this is going to be fun for me. This has been a fun challenge. I am not a doctor, and so I'm not in medicine, but I am a cell biologist, and so it's been a really fun process to read the book and think about how I can present it to you. Obviously, it's a long book, and so I can't talk about everything. But what I'd like to do is mix some of my own personal story, including my career development, some things in my personal life, my family life, weave that in with some biology and some medicine, and talking about cell therapies that figure prominently in the book. So I'm going to kind of weave all those things together. We'll see how it goes. And I hope to leave plenty of time at the end to discuss, for those of you who've read the book who want to ask more questions about various parts of the talk or other parts of the book. So I'm going to start with one slide about my research, and then I'll come back to it once during the talk just to give you a sense of why there's so many linkages with what Siddhartha Mukherjee talked about in his book and what my research program is all about. And my grad students and my postdoc are here, so I'm under pressure by them. I have to say the right stuff. Anyway, okay, so the Weiss Lab focus for my time here has been on the cellular and molecular regulation of coral algal symbiosis. So coral reefs are made up of corals, which are an intimate symbiosis between a host animal and algae that live inside of their tissues, inside of their cells, within a membrane. And so you can see that very easily here. Here's a little coral polyp and then a little tentacle that houses all these brownish algae. The brown on the reef is because of this golden hue of the algae. And this is actually one anemone cell that has about 4 algae in it. So it's a very intimate symbiosis. And my group is very interested in what is going on when everything is healthy. Healthy interpartner communication, avoiding immune destruction so the host doesn't mount an immune response to get rid of algae, and a dynamic homeostasis, which is one of my favorite parts of Mukherjee's book, is talking about homeostasis. And the immune system of the host is balanced. It's tolerating the good guys, but still healthy and able to resist negative invaders like pathogens. But we're also very interested in what happens during coral bleaching, which I think many of you know is driving the destruction of coral reef ecosystems. And in contrast to up here, we're interested in communication breakdown. The partners don't talk to one another. The holobiont, or the whole system, becomes stressed. Corals lose their algae, and symbiosis moves to dysbiosis, which is disease. So again, many linkages with the book that we'll talk about today. So that just sort of sets up why my interest in this project was so strong, because there's so many direct comparisons to what my group does. So the book structure, these are the sections of the book. And what I've decided to do is spend a little bit of time, less or more depending on what I think is the most interesting or relevant to today, on each of these sections. The first is really a history of science section, a little hard to get through in some places, but fascinating. Just the story of the discovery of cell biology. And to give something a little bit away, you can't know what you don't know. So how did people together over time, before the 20th century and even now and into the 21st century, how do they create that knowledge? Then Mukherjee goes on to talk about some basic processes like what is a cell, what is a eukaryotic cell, how does it develop, how does it divide. Then I think the centerpiece of the book, because he's a hematologist and an oncologist, there's a large section about blood, and I'll spend some time on that. There's a very short section about COVID-19 and the impact on him as a human, but on all of us. Then a section on organs, which is also a really good section. Not his area of expertise as much, but some really great chapters in there about different organs in the body. And then finally, rebirth, which is a section about stem cells, about repair, and about cancer. And so kind of a bit of a grab bag, and so we'll finish with that. So not just the linear construction of the book, but I want to give you some overarching themes about what makes the book so powerful to a an informed but not expert reader, is the use of metaphor and really vivid language throughout the book. And it was easy. It was actually hard to limit the number of quotations that I wanted to talk to you about. But also there are these overarching science themes. The first is the cell and multicellularity. Life is in general cell activity. Talking about multicellularity, a cooperative union of units, a parliament of cells. Like I've been saying that to my husband. Where is Rich? Is he here? There he is. I've been saying that to my husband for a week. He's like, let's get this book thing over with. So she'll stop talking about it. But maybe my favorite of all, the entire book, is don't just do something, stand there. That's the beautiful definition of homeostasis. I love that. That's another one I've been talking a lot about. Another theme is cell disease. So just so many metaphors. Cancer is a distorted version of our normal cells. Diabetes is cellular starvation in the presence of plenty. Sugar everywhere, but not a molecule in cells to satiate them. Regarding autoimmune disease, it's like a fire feeding on itself. Once the barrier to the self has been broken, everything that is self can be under attack. So just great language to help you understand what these processes are. And then, of course, cell therapies, which is a big focus on the book. I picked out two that are regarding the new human. The first, the new human in the title, a human rebuilt anew with modified cells who looks and feels mostly like you and me. Regarding the future treatments with pluripotent stem cells, I sometimes think of the Greek story of the Delphic boat. The boat is built of many planks. Bit by bit, the planks decay and are replaced by new ones until they are all new. But as the boat changes, even the same boat? So that kind of moves even to a little bit of philosophy. But also the book is stories, a lot of stories of people, both scientists and patients, and technological innovation, and giving you the sense that medical science, it's a human endeavor. It's, science is not objective. It's subjective because we're doing it as humans. And so to sort of break this down, these are two great quotes. A scientist, a scope, a cell. This sort of points to the individuality of science and the power of observation and discovery through observation. Not everything is about an experiment. Sometimes it's about looking. And then another quote that I've been talking about at home is an eye in a sandstorm versus sand in the eye. And this is the only quote from the book I'm going to read. And I just love this. This is about two fundamentally different ways that problems get solved in science. And this is on page 279. It might be useful to distinguish two broad kinds of problems in science. The first kind, call it eye in the sandstorm, problem arises when there's such immense confusion in a field that no pattern or roadmap is visible. There's sand in the air everywhere you look, and a completely new pathway of thinking is needed. Quantum theory serves as a good example. The second is its converse, call it sand in the eye problem. Everything makes perfect sense, except one ugly fact that just doesn't fit the beautiful theory. It irritates the scientist like a sand grain lodged in the eye. Why, she asks herself, won't this one irritating contradictory fact go away? So that's just a great contrast to the kinds of challenges that humans have in solving some of the big problems in science. But also the book is about different patients, injured World War II soldiers. And then two that I pulled out is Timothy Ray Brown, who was the first patient to be cured of AIDS by a transplant, and Emily Whitehead, who was featured very prominently as the first patient to survive a CAR T immune therapy, which I'll talk a little bit about later in the book, or in the talk. And then, of course, the scientific achievements. This gives you a sense of just how sweeping the scope of the book is. 14 Nobel Prizes are mentioned in the book explicitly, and there are actually a few more that are in there that aren't talked about directly. But each of these is a story, and if you read the book, you really read about every single one of those stories, the patients, the people, the scientists doing it. And so this gives you a sense of how important these discoveries are, how central cell biology is to medicine. One other thing to point out is, does anybody know why CRISPR is asterisked? Any ideas? Right. It is a brand name. The reason why I asterisked it, it's the only Nobel Prize that has been given to a woman of these 14. And so that's a point when we think about the discovery section, women were excluded until very recently, just societal exclusion, gender exclusion from participating in science at these really high levels. And so that's changing. CRISPR is one of the more recent, Jennifer Doudna, is one of the more recent really big discoveries in medicine and science. So anyways, just to point you out that we are victims of history, right? Women were not allowed to be at the table for many of these things. They're probably helping out, but didn't get recognition, and there are lots of stories about that. Anyway, okay, let's move on to the discovery section. And there are 2 slides for this. There are many, many stories. I thought one of the most important and really foundational stories was the story of the discovery or the development of the microscope, and how foundational that was in starting to think about cells as being really the building blocks of life. And he talks interestingly about 2 different scientists. Here I'm illustrating Anton van Leeuwenhoek, but he had a contemporary, Robert Hooke, who was a botanist, and this is actually an image of his cork that he discovered, using his microscope, which had 2 lenses, and Leeuwenhoek's, which he's holding in his hand, had just 1 lens. But Leeuwenhoek, rainy day, it's a great story, got some rainwater and stuck it on his microscope, and there are all these things swimming around. It's like, holy crap, what is this stuff? And he was really secretive, and he wouldn't share his microscope, so he was kind of weird, clearly. And Robert Hooke, and they interacted, but he was competitive and didn't want to share, so that's a great story in the book. But this is drawings that he made of the things that he found in his rainwater, and I just will point out that some of them are invertebrates. And I teach invertebrate biology, so I had to take a moment. So you can see here and here and here, these are all rotifers, which are multicellular, thousands of cells, but little tiny, tiny animals. And these are water fleas, Daphnia. There's Daphnia. And there's a quotation in the book where he's describing Daphnia swimming. And so these are things that he saw with his little microscope, but he also saw little single-celled things like sperm. So he discovered a whole cosmos previously unknown to humans. Amazing. You can't know what you don't know. We didn't know it was there, so nobody thought about it. And then the section of the book that I think sets up the discovery, that sets up the rest of the book, is his discussions about Rudolf Virchow, who he really admires, Mukherjee really admired. And he was a German pathologist who was very thoughtful. He was very focused on justice. He got involved in sort of politics at some point. But and there's a lot about him that's woven in through that discovery section. But Mukherjee considers him to be really the father of cell biology. And it wasn't just the experiments he did, but it was integrating the knowledge that he was taking from the literature as such as it was at the time and interactions with colleagues and really being thoughtful about what was going on. And so at the end of this discovery section, you emerge with really these central tenets of cell biology that Virchow posed. And these sort of set us up for the rest of the discussion. First, organisms are made of one or more cells. The cell is a basic unit of structure. All cells come from cells. This is a hilarious one. When you read the discovery section, some of the things that were thought to be causing development of humans, like little tiny fully formed humans being born. I mean, just stuff that seems ridiculous, but at the time, it made sense. You know, humors and miasmas and things like that. All cells come from cells. Normal physiology arises from normal cell physiology. And likewise, disease is disrupted cell physiology. So these central tenets are really the foundation of this book. And so if you keep those in your mind when you're reading the rest of the book, they keep coming back all the time. So next is the one and the many. This is the section sort of on some basic eukaryotic cell biology. And you can just now pretend that you're back in high school biology for a minute, and we're just going to get reoriented sort of on the basic eukaryotic animal cell, and which I just picked out of Encyclopedia Britannica clip art. And I'll sort of go through it step by step and use the, again, often very vivid language that Mukherjee uses. So the cell is surrounded by a cell membrane, which is semi-permeable, so it's not a prison. But that semi-permeable part of it is so important in biology. The cell has to control its internal environment, what gets in, what goes out, but things have to move in and out. And so there are proteins in that membrane. It's this fluid mosaic model with the lipids allowing for the movement of these proteins that are embedded in there. They're channels, they're transporters. That a lot of biology is about understanding that transport across that membrane. So there's the cell membrane. There's the captain's bridge, the nucleus, which is where the DNA is stored, the chromosomes are stored, and it's, you know, from which the nucleus is where everything's launched from, right? So DNA is stored there, RNA is transcribed there. The RNA is then transported out into the cytoplasm where things start to happen. And so the cytoplasm is the vital fluid. It's this gamish. There are lots of terms for it, but it's this sort of semi-fluid milieu of the cell itself. It also is where a lot of cytoskeleton is, so fibers that are creating structure of the cell, if it's not just a ball like this. And then how could I forget the mitochondria? Okay, so then these mitochondria are the power plants. This is where all of the ATP, which is the currency of the cell, is made. And 1.5 billion years ago, it was a symbiosis, so a free-living, and I just gave my lecture on this yesterday in my symbiosis class. A free-living bacterium was engulfed and kept in the cell, and this allowed for aerobic respiration. So 1.5 billion years ago, this didn't exist. So that was the beginning of a eukaryotic cell. Anyway, power plant. And then these different structures are super important to the functioning of what cells do. And so he talks about the protein factory, these ribosomes, which are these macromolecular structures that are either floating around in the cytoplasm or decorating the outside of this endoplasmic reticulum. And this is where proteins are made. And it's a process where the RNA is translated. I won't talk about how, but RNA is then coded and made into proteins. And then he uses the analogy of a postal service. I'm not sure I bought that entirely, but the postal service for how these proteins are moved around and trafficked into the cell. So if they're made on the edge, on a ribosome on the edge of the rough end plasmic reticulum, the proteins end up in this membrane structure that communicates with this membrane structure that eventually gets trafficked to the outside of the cell. So by a secretory vesicle, very dynamic movement of membranes that are containing proteins, and the protein gets dumped and it's going to do something outside of the cell. So that's sort of the business end of the cell in terms of making things and producing things. And then two more things. The peroxisome is sort of where proteins go to die. So this is a structure that grinds up proteins and recycles them, recycles the parts. This is important for one cell therapy that I may get to at the end. And then finally, the lysosome, also important in immunity, the cellular stomach. This is where, if there are things inside of the cell that need to be destroyed, this via membrane trafficking, the lysosome can fuse with whatever is going to be destroyed and digest it. And there are a few other things that I won't go into, but this is a complex mix of things. And so Mukherjee's comment about it is the cell is an integrating machine that must amalgamate the functions of these individual parts to enable the fundamental features of life. And this is a great way to sort of set everything up, because this cell that's not what we look at when we're thinking about the rest of the book. There are all these crazy different structures and they have all these different functions, but most cells have these basic components. And so that gets us going for different functions as we move along. This section also includes a discussion of cell division, so mitosis and meiosis. All life comes from life. And this is, how do cells reproduce themselves? And how does an organism reproduce itself? So mitosis is simple cell. Two cells. Did we die?
It looks like it died on it. Or something else is going on. Sorry to interrupt.
I'll just hold. I think I'll hold the microphone.
Oh, sure. Absolutely. Yeah, sorry for that. Yeah.
It's okay. Batteries. Thank you. Okay. Hello. Okay. Okay, so cell division. Mukherjee loves to talk about multicellularity, and he has great metaphors and language for it. This idea of moving from being a single cell to being a cooperative groups of cells. How did multicellularity evolve? And he says, we are pixelated assemblages, composites, our existence the result of cooperative agglomeration. We are a sum of parts, a life within a life. So each cell has its own life within this larger life. And I just put here a great model for the study of the evolution of multicellularity, which is this choanoflagellate, which are these little single-celled organisms that are at the very base of the animal tree that sometimes just decide they want to be together. And once they're together, their properties change and they start to cooperate and all sorts of very interesting things happen. But they can also revert back to the single-celled stage. So that's a model system that's commonly used for multicellularity. And then there's a section on animal development, which that's Barb's field, by the way. Barb was a developmental biologist before she retired. So, how do you start with one cell, a zygote, and end up with a whole human? What are those processes? The way in which cells divide and move past each other and end up positionally makes a huge difference as to what that organ or that tissue will be. And so life not so much is as becomes. So it's a process, and that's developmental biology. And this just illustrates just a schematic of what a mammalian blastocyst, very early stage past a zygote, and this is the future embryo, and this is the future placenta, and this is an actual image of a blastocyst, and you can see that little shelf that Mukherjee talks about. Okay, moving on to blood. As I said, this is really the centerpiece of his book because this is what he does. He's a hematologist and an oncologist. And he calls blood a liquid organ. I really love that because it's a liquid, but it's so complex. And the other part I liked about this section was that he really shows some personality. You get a sense, he shows who he is as a person when he talks more about areas that are close to him. And he talks about his typical Monday when he's allowed to be a scientist again and allowed to be just looking at cells instead of doing all the crazy stuff he must be doing. A droplet of blood has been spread across a slide to make a film of single cells, each stained with special dyes. The slides are like previews of books or movie trailers. The cells will begin to reveal the stories of the patients even before I see them in person. And then he says, it is by far the favorite time of my day at work. Number, color, morphology, shape, size. He's going through what he learned as a student. These are the things I'm looking for to try to help this patient, try to diagnose this patient's disease. And so this is a typical human blood smear. And in the next slide, we'll talk about it too. But you can see that by far the most cells are the red blood cells, are cells that are transporting oxygen. But in amongst here are a few white cells, white blood cells. And then the little bits are not a mistake. Those are the platelets. And so those are the clotting factors that prevent us from bleeding out when we have a cut. So this is what he likes to spend his days doing, but he only gets to do it on Monday. But I really wonder, does he really do it every Monday? Because he seems like a busy guy. Okay, so then he goes on to talk about blood. In fact, blood speaks to everyone and everything. It is the central mechanism of long-distance communication, of transmission in humans. Be it hormones, nutrients, oxygen, or waste products, blood delivers and connects, talks to every organ and from one organ to the next. It even speaks to itself. Its three cellular components, red cells, white cells, and platelets, in particular, engage in an elaborate system of signaling and crosstalk. So much going on there. And we'll just briefly cover it here. This is when you go get your blood drawn later on, or if you just sit it on the counter, the red blood cells will pellet because they're heavier. There's a little layer here, if you spin it, that's called the buffy coat, which is where all the white blood cells lie. And then the rest of it is plasma, the fluid of the blood that is containing all of these things that he talks about here. And so he devotes many chapters in the book to these different components, talks about erythrocytes in one, platelets in another, and then I think 3 chapters on the white blood cells. But I will note that with regard to the red blood cells, there's a very recent development that you probably all know about. He talks about sickle cell disease at a couple of places in the book. And since the book came out in 2022, and he was probably writing it in 2021, there is dramatic news that sickle cell disease treatments have now been approved that are cell therapies, right? One of them is a CRISPR. There are other other important therapies that are aimed at basically curing a patient by changing their genetic structure. And sickle cell disease is caused by hemoglobin that is mutated and it stacks up, and so it contorts the shape of the red blood cell and causes extreme pain. It also causes, they don't transport oxygen as well. And so the idea that this disease can be cured, it's very exciting, albeit not scalable yet because it's so expensive. But this is a great update from when the book was written. And then let's talk a little bit about the immune cells. These are the white blood cells. And he devotes 3 chapters on lymphocytes, and then 1 chapter on sort of the mixture of the rest of them. And so what I'm going to do, and so these are the cells that are really the immune system. There are other parts of the immune system too, but these are really where the business end of immunity in humans is. And so let's talk first about the guardian cell. He calls them innate first responders, contortionists. This is because many of them are phagocytic and they contort and they could burst through blood vessels, move into tissues, roam around. They could roam all through the body. And they're professional killers. And so these guardian cells are part of innate immunity in humans. And innate immunity is a very ancient, highly conserved process that is quick, doesn't have memory, and is usually responding to a common limited set of markers on microbes or abnormal cells. They recognize these markers and then they launch either killing or various other ways to control that disease. So, for example, pus, if you cut yourself and then you get your cut is infected, that pus is the battlefield. These cells have gone there to kill bacteria, ingest it, destroy themselves. And so that pus that you see is thousands of dead bacteria and dead human cells that are, soldiers killed in battle. So these phagocytic cells, they're actually called professional phagocytes. That's the general term for them. And what they do is they detect a key on this pathogen because they have a set of locks that are waiting to sense, is there something there? Is this negative? Is it bad? Is it bad? If it's bad, oh my gosh, I need to eat it. And then what happens is they undergo phagocytosis. They suck, they swallow up the microbe by containing it in a vesicle. And then that lysosome, the cellular stomach, goes in, fuses, and destroys it and spits it out, or the whole cell will die. And so these professional phagocytes, this is what they do. This is their job. And they're roaming around all the time in your system, throughout your body, moving through the blood, and then bursting through to other places, and they're doing this. This is where there's a tie to the Weiss lab. So corals have microbe-eating cells. Remember, I said that it's an ancient, highly conserved process. And so this is a high magnification of a little baby coral that has been fed algae that it's going to become symbiotic with. And look, it's reaching up with its pseudopods just like a professional phagocyte, and it's going to engulf that alga, just like this human macrophage, which is eating a little yeast with a little face, little eye, nose, mouth, going to get sucked up. Same set of mechanisms used, same genes. So my joke about cnidarians is humans are just smart cnidarians. Same set of genes. We have a very highly conserved set of genes for these kinds of things. But the outcome is different. In corals, the algae persist because that's the right answer. The algae persist. They've either evaded the host or the host recognizes them as friend, and they persist. This is a little baby coral, little larva, and the algae don't get digested. Somehow this digestion process is blocked and they persist inside of the host. And so the interest in this part of the book is obviously piqued for me because it's what we study. So the next thing I'm going to talk about is the defending cell, B cells and adaptive immunity. He has 2 chapters on T cells, super complex. Not really enough time to try to simplify it and go through it step by step. But I can talk a little bit about adaptive immunity with B cells. So adaptive immunity is present in jawed vertebrates only, and it's that memory part of immunity. It's what gives us vaccines. And so I'm just going to give you a sense, and he goes through this in the book. I'm going to give you a sense of what a B cell does. So in our bodies, we have up to 1 million unique B cells with these antigen receptors, future antibodies, just hanging out, roaming around, okay, waiting to interact with some sort of antigen, our microbe with an antigen on it. with something sticking out that they can recognize. And so one of these B cells will bind this antigen and say, oh my goodness, something's happening. I need to become activated. So these B cells get activated, and then they start to clonally expand. So this single cell expands into many, many cells. This process can also be caused to happen by these helper T cells. He spends a lot of time talking about them. There are 10 million unique helper T cells in our body. So there's this magnification process that allows for the B cells to be activated in more than one way. Clonal selection happens, then differentiation happens, where this clone goes into two types of things. It goes into memory cells, and it differentiates into plasma cells. And the plasma cells, I'll show you in the next slide, become antibody factories. Their job is to churn out the antibodies. So they make rough endoplasmic reticulum, traffic it out, dump antibodies into the bloodstream. Anticorpor is an antibody. So here are the antibodies now floating around in the blood. They find a microbe, they attach to the microbe, then a macrophage from that innate immune system, professional phagocyte, sees that an antibody's been bound, binds the antibody, and destroys the macrophage. So that's what's happening with memory or with innate immunity, is that the whole immune system participates, but the memory is held in the B cells themselves. And it's these memory cells that exist indefinitely in your body that allow for the development of vaccines. Because if you use a little bit of compromised microbe up there, a little bit of compromised COVID, right, then your body mounts a response and it's ready the next time that you get infected. So this differentiation, let's talk about this for a minute. Here's a B cell. This is a transmission electron micrograph. Here's a plasma cell. Look at the rough endoplasmic reticulum. This is a dramatic morphological change in the cell, and they're just cranking out the antibodies and dumping them into the bloodstream. And this is a key place for one immune therapy that he talks about. And lymphoma is a cancer of B cells. So this is one blood cancer that is common. And all B cells express CD20 on the surface. So these flags, as he says, flags on the surface of B cells, but they're absent from plasma cells. And this difference was used for the first immune therapy or antibody therapy ever development, where they used an antibody that they manufactured in a certain kind of mouse cell to target the CD20 so that you could target the cancer but protect the plasma cell. And so this is what the inventors said: we could fight fire with fire. So use the very system of antibodies and memory to attack the cancer. There are now about a dozen of these antibody therapies for blood cancers. They've yet to work well on solid tumor cancers. He then has a sort of an interlude about the pandemic. And he was writing the book during the pandemic, so he really had to find a way to, well, obviously it affected him, but find a way to talk about it in the context of the book. And he says, I cannot shake the images or the sounds of the pandemic. Who can? His perspective on the immune system or its field before COVID is, he was talking about a colleague. He told me that researchers were increasingly imagining the immune system as a knowable machine with movable, manipulable, decipherable, changeable cogs, gears, and parts. I did not sense hubris in his optimism. So he was saying, before COVID hit, we kind of thought we had it. We had this thing down, like we'd figured it all out. And yet, SARS-CoV-2 is a dysregulation of immune cells at its core. The pandemic is a disease of cells too. And regarding the cytokine storm, which you all heard about, the out-of-control immune response, but as the house burned, the cell yanked the trigger on a powerful second alarm, sending out a separate series of danger signals, cytokines, to summon immune cells. An uncoordinated army of cells, confused, bamboozled soldiers surged to the site of the infection and started a carpet bombing campaign. Too much, too late. The trigger-happy immune cells poured out a fog of toxins to contain the virus. The war against the virus, as much as the virus itself, became an escalating crisis. So in the early days of COVID, the cytokine storm, they couldn't control it. They couldn't figure out how to help the patient survive that. And he talks a lot about why is all this stuff happening? We don't know. Why long COVID? We don't know. And then he talks about this Donald Rumsfeldian comment, the known knowns versus the unknown unknowns. And my lab groups here, I can say to them, I will often say to them, you can't know what you don't know. We only knew COVID based on the knowledge that was in hand at the time. There is undiscovered information out there that could have helped, but it's not yet known. And so I think that was really the point of the chapter, is we don't really know everything, and we need to be humble about that. Okay, I feel like I'm running a little short on time, so I'm going to try to speed up. So the next section is on organs. There's a great section about homeostasis, which I didn't really decide to develop, but I want to just focus on the nervous system that he featured in one chapter, and feature the illustrations of Cajal, a neuroanatomist who actually received a Nobel Prize in part for describing the neuroanatomy of vertebrates. And there is a great story about him disagreeing with another guy, and they shared the prize, and they were like, do we have to share this? Because we don't agree. But he believed in the nervous system based on a series of wiring and not just some random array of lacy things, which is what Golgi thought it was. But these are incredibly beautiful drawings, and Mukherjee points out they're one of the most beautiful science art in existence, and I totally agree. But I will say this drawing in the book is mislabeled. It is not the brain, it is the retina. And I recognize that, and I'll tell you why in a minute. But the synapse was the topic of the sand in the eye problem, is they couldn't agree about this whole weird thing about the synapse. Why wouldn't wires touch each other? That's a very interesting sort of side story. But this retina, when we look up close to it, it goes back to my light bulb moment as a little undergraduate college student. And when I was a junior in college, I was given the privilege of being in a cell biology lab where we would learn electron microscopy. There I am with my friends in 1983 when I was a junior, looking very bright-eyed and bushy-tailed and very non-gray. And it's this electron microscope that was developed by Palade and Sikowitz in the 1960s, only 20 years before I was privileged to be able to do this. And this machine is the size of a room. So imagine being like a 20-year-old and being able to go into this room with this giant machine and being allowed to touch it and work it. Oh my gosh, I was hooked. And my tissue was mouse retina. I got to study mouse retina. And this is what it looks like. Just these long rod cells with these stacks of membranes where all the pigments are that allow you to see. And then these cells in the back that absorb extra light. I used to see them in the shower. I mean, I literally was like, I was hooked. And so that was the beginning of my science journey. A scientist to scope a cell. I didn't know it at the time, but I was a budding scientist. Okay. Stem cells. He has a whole section on rebirth, and he talks about how through development, if you start with a blastocyst, you get to these different tissues and cells and tissues. All these dramatically beautiful and different tissues are coming from originally a single cell. That at least was understood. But the idea that we might be able to work with stem cells that could drive this and develop into these different tissues was unthinkable. No embryologist that I knew thought of embryology as a two-way street. Go forward and you get a human, go backward and you get a mature cell, nerve, blood, liver, and turn it into an embryonic stem cell. It seems like madness, one researcher told me. And that's exactly what happened in 2006. Yamanaka in Kyoto, he took skin fibroblasts, which are just garden variety skin cells, determinate, right? They're done. They're not going anywhere. They're not going to make anything else. And by manipulating them, he caused them to become stem cells. The production of induced pluripotent stem cells was an utter shock to biologists, a loma prieta that shook the earth plates of the stem cell world. And because of this, he received a Nobel Prize. He did it first with mouse, and then he did it with humans a year later. And so these pluripotent stem cells allow for potential stem cell therapies in the future for all areas of the body that where new tissue is desired, including, he talks about diabetes and the pancreas. And just a quick note that pluripotent versus multipotent terminology, but multipotent is, for example, the B cell is kind of multipotent. It can make a type of the plasma cell, but pluripotent cells can make virtually all cell types in the body. So then finally cancer, I'm running out of time. Cancer, he talks a lot about. He's written a whole book that won a Pulitzer Prize about cancer, The Emperor of All Maladies. Cancer is a disorder of internal homeostasis, a jammed accelerator, and snapped brakes. Natural selection creates the cells that disobey every law of belonging except for the laws that they have created for themselves. These cells break the laws of cellular citizenship. And the playlist is set. The same series of songs get played over and over again like a malevolent tune that one cannot get out of one's head. And when you apply a selective pressure, such as a drug or immunotherapy, it switches to a new list of genes or even scrambles the songs, remixing a crazy jumble of hip-hop and Chopin, say, in its playlist, such that the malignant cells can escape the drug and then repeat. Now the cancer cell has new fixed malignant tune that it cannot get out of its head. And I'm going to tell you a quick story about plasma cells. Remember, those are the antibody factory cells that produce antibodies, so they're just made to produce antibodies. Multiple myeloma is a cancer of plasma cells, and so it's a malignant stem cell plasma cell that starts to make antibodies for nothing and just starts dumping them into the bloodstream and growing like crazy. And so really what you get is you get bone marrow full of cells, and you get tons of antibody everywhere that has no meaning in your body. Multiple myeloma causes bone breaks, it causes anemia, and it causes kidney failure because all of these proteins cannot be cleared and they end up clogging the kidneys. And I want to talk about stem cell therapy for multiple myeloma, and it has a very personal story. My late first husband, Mark Patterson, died in 2006 from multiple myeloma. Here he is in 2002. He is not a mountain man. He's actually a father on the teacups with my kids who are now 30 and 28 before he was sick. But he had multiple myeloma, and the section in the book about the Fred Hutchinson Cancer Research Center is very near and dear to my heart because this is where Mark went to receive autologous stem cell therapy. So he had not an allogeneic, not a donor transplant, but cells from his own body were taken out, cleaned up, and put back in. And this bought him about 2.5 years of life. So he was diagnosed in 2002, was treated, and then lived for a few more years. So what happens with this is they harvest stem cells, and then they freeze them, and then they give the patient a giant chemo dose, killing all dividing cells in the body, making, of course, the person very, very sick. And then they re-infuse the cells back into the body, and the bone marrow gets to repopulate with stem cells that are less cancerous and thereby buying you a few years. But I want to point out that now the Hutch is focused on immunotherapy. So there are all these new therapies out for cancer, and it's really a hopeful day for myeloma, which is a very rare disease, but also other blood cancers. There are drug treatments. There's antibody therapy, just like the therapy that I talked about with B cells, but different antibodies. There's CAR-T immunotherapy, and there are other therapies under development. So the prognosis for myeloma is much better than it was when he died almost 20 years ago. So, the poignancy of the nurses that he describes in the book going through the difficult early days of transplant science and discovery really spoke to me. It brought back a lot of memories. Okay, final slide. He finishes talking about the songs of the cell. So to start again with sort of Virchow's tenet, cell physiology is the basis for human physiology, and cellular pathology is the basis for human pathology. To build new humans out of cells, we need knowledge that is not just names, but the interconnectedness between names, not identification cards, but personalities, stories, and the histories that accompany them. We've learned about cells, systems of cells, organs, and tissues, but there is yet another layer of organization left to learn, the ecosystems of cells. It is the music that drives the complexity of cellular physiology, and conversely, the playlist of malignant pathology that remains one of the unsolved puzzles of biology. We can name cells and even systems of cells, but we are yet to learn the songs of the cell, sort of that meta level. And that's where he leaves the book saying, this is what we still have to learn. And so I'll stop there.
Great.
Yes, happy to take questions or comments.
Yeah, I don't have a question like that of how that film was like people science education biology. Sorry, books like this it's fascinating. But my comment is really wonderful. Now, sick reading for that. Thank you very much.
Thank you.
Has made a better sure around it will be a better.
Thank you so much. Yeah, there's a lot in there. There are many stories. I could make an entirely different presentation with a different set of stories. So I had to pick and choose, but thank you very much. Yeah.
John, you probably start what they tell us.
So the RNA vaccines are really different, and I won't talk about them here, but basically, if I understand it correctly, every time they release a new vaccine, right, they've changed that antigen a little bit because they have new information about what the COVID surface looks like. And of course, it's rapidly evolving, and this is why we have to keep getting vaccinated. And so they'll update it based on their best knowledge. And so the antigen will be slightly different. And so it will be a different set. It will be a different set of B cells. So the B cells that are in memory of all the other vaccines we've had, there'll be a new set started that will help us fight future different COVIDs. Yeah, great question. Oh, hi, Jenny. Yeah.
So, do you think
Yeah. Parts of it are tough, honestly. You really do. It helps to have some background in biology. I think the discovery part, you don't really have to. because it really is a history of science. And then parts of it, like the section, which I didn't get to talk about because I ran out of time, on homeostasis, where he talks about a dinner in Venice, and you have wine with your dinner. And so then, the kidneys have to work on salt balance, and the pancreas has to work on sugar, and then the liver has to detoxify the wine. That is an awesome section. So those parts, but then when he, like the blood part, which I liked a lot, but I know a lot, right? There's a lot of nerdy stuff in there. So I think it's a mix. Yeah. So I think it's a little uneven in that I think you could get bogged down. And so I guess for those of you who hadn't read it who were interested, if you start to get bogged down, you could just move on to another section. It's not necessarily contiguous.
Comment. Actually,
Thank you. I've never done this kind of thing before. So the whole quotes thing, like you don't do that in science. So that was challenging. But once I got into it, then I was like, Virginia, pull back. I had to take a bunch of stuff out. So thank you. Oh, yeah. Yeah, I can try. So CRISPR is, the biology of it is fascinating. It's a process that's been evolved in other organisms. But basically, it's a way to edit genes, okay? And so it's not just one thing, but there are these guide RNAs. So there are these bits of RNA that can be put into a cell with the correct protein that will sit down and recognize, okay, that hemoglobin in sickle cell patients, that hemoglobin's messed up. So I'm going to design this guide RNA with this particular protein that has to go in there that's going to find that gene, find that broken bit, and put in the right, correct, normal, healthy bit. And so that's the way it can be curative, because you can eliminate the cells that have the mutated hemoglobin and put in cells that have the right one. So that's it is hard to understate the importance of CRISPR in biology. So my lab, my student Olivia, she is determined to get CRISPR going in my group, which is exciting, and it has started to happen in some coral work. There are challenges, but it's hard to understate how important it is for biology and for medicine. Yeah. Oh, Leeuwenhoek. Yeah.
I was like that the other so fast, but doing job sharing how that he that's like oh
Yeah.
that no one can not walk out that
Yeah.
and he saw that all that no one that like how could I
Wow, boy, put me on the spot.
right.
Oh, yeah. Yeah. No, that's why I thought the discovery section was so good, is that it was sometimes hard to get through, but there were some of these aha moments, right? And honestly, CRISPR is another one of them, because it's not just really cool biology, but it's so foundational. I mean, the structure of DNA is another one. It's just like, wow, that just changes the whole thing, right? And just think about like Gregor Mendel, the geneticist who was a monk in like 1602 or something, who didn't know anything. And he discovered all this stuff. And then you go back and you look at it, and it was all because of this DNA. So there are lots like that. The Leeuwenhoek one is so interesting because he was a weirdo. He was a cloth merchant who actually developed the microscope so we could look at the quality of the threads of the cloth. And then he just is like, one rainy day, he's like, maybe I'll just take a drop of water. Oh my goodness. So, and then, and then he tried to publish, and they're like, okay, well, share your microscope. No, I don't want to share. I'm not going to share. So, very strange and very, very interesting stories. Oh, it's coming. It's coming. Not right away, but soon. Yeah. Okay. All right. Thank you all so much for coming.
We're in the house.
I do okay.
Let's go.